Hello students, welcome to LEELA'S tutorial. In last class we have discussed about androgenic haploid plant production. Right? So today let us see the production of haploid plants from the female reproductive system. That is, by using the gynecium, how you are going to produce the haploid plants. So again, let us see what is the structure involved in the male and the female reproductive system of the flower. Here this is the stigma, style and ovary with ovules. Right? It can be a single ovule or many ovules. Right? So which later transform into seeds. Then you have this male gamete, which is the anther and the pollen grains. You can see the filament, anther with pollen grains. In last class we have produced the haploid plants by culturing the anther or the pollen grains, and they are referred as androgenic haploid plants. So today we will produce the plantlets, that is the haploid plantlets from the female gametes. This is the male. So from the female gametes, when you produce the haploid plants, they inherit only a single set of chromosomes from the female parent. Right? So what is the explant you are going to use from this gynecium? It is either the complete ovary or the ovules. If it is a single ovule, then you go for the ovary culture. If it consists of many ovules, like in case of pomegranate, it has many ovules, right? So in that case, you will collect the ovules and you go for the ovule culture. Okay? So here, the gametes are haploid, which consists of only one set of chromosomes from single parent, right? So here all these cells present in the animal or the plant is a diploid. The somatic cells are always diploid. So what are haploid cells in an organism? The haploid cells are the gametic cells. So here the gametic cells, they are formed by the reduction division, that is by means of meiosis. The 2n number of chromosomes which are present in the diploid parent are transformed to haploid, or only one set of chromosomes are present in the gametes. So here, without fertilization, if the male and the female gametes, right, are getting fertilized, it will result in the formation of diploid zygote. That means the single set chromosomes, that means n number of chromosomes present in the male gamete, that is the pollen grains, will fertilize the gynecium. Again, here it consists of only one n chromosomes, that is again it is a haploid. Right? The male gamete and the female gametes are haploid. When they undergo fertilization, they result in formation of diploid embryo, which transforms into a fruit. Then the seeds which are present, the fruit or the seeds which are present are diploid and give rise to a diploid plantlet. Whereas here, today we are going to make use of only the female reproductive system, that is the gynecium. We are going to collect the ovary or the ovules before fertilization process. Right? So here you must take the closed bud so that no pollen grain has entered into the style of the gynecium. Okay? So you have to take unfertilized ovary or unfertilized ovules. If you are taking fertilized ovary or the ovules, it will result in diploid plantlets, but we want only the haploid plantlets. So why we must go for this haploid plantlet production when we have natural mode of pollination process? It can be a wind pollination or by insects or anything, right? So what is the use of this production of haploid plants in plant tissue culture? The main use is here in some of the plants, the natural way, though the pollination occurs, though the pollen grains enter into the stigma, style and the ovary, it fails to fertilize. Right? So it will fail to fertilize, so that those plants will fail to produce a fruit or seeds. Right? So in order to propagate that, in order to undergo the propagation of those plants, we make use of this plant tissue culture process where we are going to produce the haploid plants. Right? So here in plant families that do not respond to the androgenesis, we go for this gynomorphic haploid production. Right? The haploid plants, if they are produced by using the female reproductive gamete, then you call it as gynogenic haploid plants. Let us see the technique. Here in this process, first you are going to select the disease-free plant, right? The mature healthy plant, and you are going to select the flowers and the flower buds in such a way that they have the mature ovules or the matured ovary. Okay? We are not selecting any flower, but we are selecting the flower bud, so that it is sure that they are maintained under aseptic condition inside the closed petals, right? And also there is no pollination has occurred, right? So we want unfertilized ovary or the ovules. First collect the flower bud, then go for the surface sterilization by using your mercury chloride or sodium hypochlorite or silver nitrate, any one of the surface sterilant, and then you are going to remove all the dust and the contaminants present over the surface of the flower, right, of the flower bud. Then after that you are going to tear open the flower bud in laminar airflow. Place this in a petri plate, right? In a sterilized condition in laminar airflow, you are going to cut open the flower bud and collect only this portion. Either the ovary, if it consists of single ovule, if it consists of multiple ovules, that means if it consists of like this ovules, then you are going to tear open the ovary and collect the ovules. Is that clear? If it is a single ovule, you are going to culture complete ovary. If it is many ovules present in that, then you will take the ovule. You will tear open the ovary and collect these small ovules. Right? So here after surface sterilization, isolation of the ovule or the ovary, then inoculation. See here I have shown here it consists of, it is a complete ovary. It is a complete ovary consisting of many ovules. Whereas here it is a ovary consisting of single ovule. Right? So in this case you will directly inoculate that into the plant tissue culture media. In this case you are going to tear open or cut open the ovary and collect those small ovules, and only the ovules are inoculated. Right? After inoculation of these ovules or the ovary into plant tissue culture, you are going to incubate it. Right? So incubate it with the required temperature, time, pH, all the environmental conditions along with the nutrients. Right? So when you provide sufficient amount of nutrients and all favorable environmental conditions, here now the ovule or the ovary withstand in the media and they respond to the media, observe the media, and they get started with the cell division. Right? As a result, they transform into a callus. See here, the ovule or the ovary culture, they can be done in suspension culture or semi-solid culture. That is the liquid media or the semi-solid media. Mostly if it is a ovule culture. Ovule culture, it is very difficult to handle the very small ovules. In that case, we go for the suspension culture or the liquid culture. In case if it is a ovule which is bigger in size, then it is easy to handle with the forceps, right? So you can transfer that on the semi-solid media. So again, with the required amount of hormones, auxin to cytokinin ratio 1:1, it will result in development of callus. Callus regeneration occurs. Callus is nothing but mass of undifferentiated cells, right? The unorganized mass of cells which have ability to transform into a complete plantlet, right? They have the totipotency effect. So now these callus under cultural conditions, right? Again here you are going to refer the embryo culture topic, right? How you are going to culture the embryos or produce the embryos from the callus. Refer those previous classes so that you can see how the embryos are generated from the callus. So once you get the embryo, right, either it can be a globular shaped, heart shaped, or the torpedo shaped. Now these embryos will, when inoculated in plant tissue culture media, they develop into a haploid plants. So now these haploid plants can be transferred to the soil, and these are referred as gynogenic haploid plants because they consist of only single set of chromosomes which are inherited from the female parent. Right? Only the female gametes, ovules, has inherited the chromosomes to these plantlets. Is that clear? If you have any doubts, you can put it in comments and subscribe for further videos.